Bismillahirrahmanirrahim. Assalamu alaikum everyone. My name is Abdulaziz Alaklabi. I'm a third year medical student, and today we'll be covering fatty acids oxidation and ketone bodies. Okay. So these slides are all from Dr. Abdul Jabbar. They're not from me. So I'll just be covering them, inshallah. So the objectives today, we'll talk about mobilization of stored fatty acids. We'll talk about then the mechanism of triglyceride degradation in the liver and adipose tissue, mainly adipose tissue. The oxidation, which is the main key point or objective of this lecture, as well as ketone bodies and their use. So before we start, please know that this lecture is for you guys. Anytime you guys want to stop me, go ahead. Any questions you guys have, you guys have to ask that. I'm open to any questions. We can stop and answer any questions you guys have. So to start off, basically here, the body fat is just our major source of stored energy. That is the major source of stored energy in our body. And in adipose tissue, we have triglycerides. Triglycerides are everywhere in the body, but mainly stored in adipose tissue. Triglycerides are made of two components, glycerol and 3 fatty acids. We know glycerol can be used in gluconeogenesis or glycolysis because it can convert into one of the intermediates in that glycolysis or gluconeogenesis stage. And then free fatty acids, as we're going to talk about today, are oxidized to what we call acetyl-CoA. And this is through beta oxidation. And this can give us much more energy, which is why fat in general is the main source of stored energy in the body, especially during prolonged fasting, as we will see. So this slide, I think you guys have already seen this in much more in depth in the lipoprotein lecture. So you guys probably know much more details than what I'll be covering today. But I like this graph because specifically here in the key, we can see the different components of the lipoproteins from this type of key, you could say. So we see in the chylomicrons, they're minimal blue, means they only have a little bit of protein. They have a lot of triglycerides and a little bit of cholesterol. We can see in the HDL, it's high in protein and high in cholesterol, but very minimal, in triglycerides amount. So I like this picture because it gives you kind of a greater picture of the composition or the circulation of fat and cholesterol. So we can see here in the diet, after you eat a meal, dietary fat and cholesterol is mainly in the form of chylomicrons, okay? And as it goes into intestine, chylomicrons, I hope you guys know this from the other lectures, it will go in the lymph first and foremost. And this is because of the certain receptors that they have. Hopefully this was covered in your other lectures, like ApoB48. I think you guys may know this. Therefore, chylomicrons go to the lymph first and foremost, and then into the circulation. In the circulation, after it reaches capillaries of the adipose tissue, we have something, I'm pretty sure you guys have all heard this, lipoprotein lipase. Now, lipoprotein lipase, what it does, it hydrolyzes the triglycerides in the chylomicrons and stores them in the adipose tissue. Now, once this process has finished, most of the triglycerides have been taken away from the chylomicrons. Therefore, the chylomicrons will be called remnant chylomicrons because they've lost most of their triglycerides. These remnant chylomicrons will return to the liver, and the liver will break them down and will take the fat and cholesterol to create another lipoprotein, which is VLDLs. Okay, so this is what the liver creates from the fat and cholesterol to send to the rest of the body. Now, VLDLs also go through the same mechanism where they are hydrolyzed by lipoprotein lipase, and then they get transformed to IDL and the rest of the sequences. I want to touch up a little bit about this. Don't want to go really in depth because our main point is over here, where we want to talk about the hydrolysis of triglycerides, storage, and mobilization of them. So, okay. So when we talked about chylomicrons and when they reach the capillaries, they are hydrolyzed, as we said, by lipoprotein lipase. So this is going to give us an in-depth picture of this. So we can see here, this is the entirety of the chylomicron. The chylomicron has a specific, what you call receptor or component that attaches to the lipoprotein lipase. This is called ApoC2. Now, ApoC2 activates lipoprotein lipase. So without it, it would not be active. It would not be in an active state. So lipoprotein lipase requires ApoC2 to activate it. Could you please use the point? No problem. Is that better? Okay. Yes, Noor. ApoC2 is on the chylomicron itself. And essentially, it is what activates lipoprotein lipase. It attaches to the lipoprotein lipase and activates it. So essentially, what we see here is the binding of the chylomicron to the lipoprotein lipase. It basically anchors on the chylomicron and then hydrolyzes the triglycerides. And the triglycerides will be broken up to, as we said, 1 glycerol and 3 fatty acids. And they will enter the adipose tissue and basically reform and be stored as fat. And we can see here they're activated by apoprotein C2. So this is very important. The lipoprotein lipase is activated by the ApoC2. So before we go to this slide, I want to go over here first and foremost, because this gives you a bigger picture, then we can go in depth to each part, okay? So we talked about essentially the storage. So basically the chylomicrons will go into the capillaries and the lipoprotein lipase will hydrolyze the triglycerides and they will be stored in the fat. I think there's a question. Okay, no problem. So it will be stored as fat. So now we want to mobilize this fat. So if you're in a state of starvation, you have no more glucose from the diet. You've finished all your glycogen storages and gluconeogenesis occurred, but your body still needs energy. You're in a prolonged fast. Therefore, the body will use the mobilization of free fatty acids as energy. So this is the point of what we're trying to cover right now, is the mobilization of the fatty acids from the adipose tissue. So first and foremost, when you're in a state of starvation, you don't have high insulin, you have high glucagon. So this is very important because the two are have opposite effects. As we can see here, glucagon will activate adenyl cyclase. This will increase CAMP levels. Now the CAMP will activate protein kinase A. This is in basic biochemistry steps. There's nothing special about this. CAMP activates protein kinase A. However, in this case, protein kinase A will do multiple things, okay? Very important. So first and foremost, as we'll see, it activates what we call hormone-sensitive lipase. We're gonna go in depth, inshallah, to each step, but we want to talk about what, first of all, protein kinase A does. So protein kinase A does multiple things. So first and foremost, it activates hormone-sensitive lipase. It phosphorylates it and puts it in an active state. That's number one. Number two, it will activate something called perilipin. Perilipin will be phosphorylated and will combine with hormone-sensitive lipase, and they together as a complex work together, okay? So now we had two things. We had hormone-sensitive lipase being phosphorylated and perilipin. So that was the first two things that protein kinase A did. When we phosphorylated the perilipin, as we can see here, what happened was there was a complex. It was a CGI perilipin complex. It dissociated. So once we phosphorylated the perilipin, it dissociated from the CGI, okay? The perilipin went to the HCl, or the HSL, sorry, the hormone-sensitive lipase, and they act as a complex together. The CGI, which is not phosphorylated, by the way, it will go what we call adipose triglyceride lipase. So we have two separate complexes, which is the adipose triglyceride lipase with the CGI58, and we have hormone-sensitive lipase with perilipin. So basically, protein kinase A activated essentially two lipases in one step. That's what it did. So it phosphorylated the hormone-sensitive lipase, and then it phosphorylated the perilipin. When it phosphorylated the perilipin, it caused dissociation from the CGI. The CGI went to bind and activate the adipose triglyceride lipase, and the perilipin, which is now phosphorylated, it will go with the hormone-sensitive lipase, which will act as a complex together. So after this step, number 3 and 4, Faisal, it's always glucagon associated with CAMP and insulin associated with phosphates. I think you mean phosphorylation. Faisal, this is in most cases. This is not a general rule. When I meant this is basic biochemistry, I mean that CAMP activates protein kinase A always. Glucagon, in some states, some states in the body, it will act something different. They will still have opposite effects. However, in this specific case, it's completely opposite, where glucagon activates CAMP and insulin will phosphorylate the hormone-sensitive lipase, okay? Or dephosphorylate it, sorry. Okay, no problem. So after step 3 and 4, we're talking about now, the pKa has now phosphorylated the two things. We talked about hormone-sensitive lipase and the perilipin. So now the 3 lipases, we have 3 lipases in the mobilization of fatty acids. We have adipose triglyceride lipase, which basically degrades the triglyceride into a diglyceride. And it's from the name triglyceride lipase, it will affect the triglyceride itself. And then the hormone-sensitive lipase, it degrades essentially the diglyceride into a monoglyceride. And then we have a third lipase, which is the monoglyceride lipase. So essentially, after all these steps, these 3 lipases will then break the triglyceride into a glycerol and 3 free fatty acids. And then these fatty acids, as we can see in step number 9, it will go in the blood and will bind to albumin. Albumin is what will take it to other parts in the body. Insulin, we'll get to that right now, Noura, okay? So did anyone not understand these steps right here? Before we're gonna go to the other slides and go in depth a little bit. Okay, great. So let's go back to the detailed slides. So this is again talking about those 3 lipases that we talked about. So you can see right here, lipases break down the triglycerides, of course. Glucagon and epinephrine is very specific. They activate hormone-sensitive lipase. So glucagon and epinephrine will activate the hormone-sensitive lipase, while insulin will deactivate this. This is very important. Can you repeat 5? No problem. So number 5, this is simply the dissociation of CGI from perilipin. So we said pKa, protein kinase A, will phosphorylate 2 things. It will phosphorylate the hormone-sensitive lipase, which is the second lipase in the steps of mobilization of fatty acids. And then it will also phosphate or put a phosphate on the perilipin. When this happens, there's basically a dissociation. Perilipin was bound to CGI. Once the pKa phosphorylated the perilipin part, it dissociates. The CGI will go to the adipose triglyceride lipase, and it requires CGI to be activated. That's all it is. So these 2 will dissociate. One will go to the hormone-sensitive lipase, the other will go to the adipose triglyceride, okay? Did that answer your question? Okay, no problem. Okay, so going back. Okay, so the glucagon, as we said, glucagon and epinephrine, very important that they activate hormone-sensitive lipase. So that means glucagon will help mobilize fatty acids. Insulin, this is how I like to think about it. Insulin is what we call the storage molecule, okay? So it allows glucose to be stored, allows fatty acids to be stored. Glucagon, on the other hand, it will mobilize everything. It will cause gluconeogenesis. It will cause increased glucose. It will cause increased free fatty acids. So it's the opposite effect. So insulin will inactivate hormone-sensitive lipase. This will inhibit mobilization of these free fatty acids, while the glucagon will actually do the opposite and will cause mobilization. It will activate hormone-sensitive lipase and will activate the degradation of the triglycerides. So, as we said right here, the free fatty acids are then exported to the blood, and they are bound to albumin. They need to be bound to albumin because they're just not hydrophilic. That's just all it is. As well, the glycerol is also released to the blood and taken into the liver cells and used for gluconeogenesis, simply. So this is what we're talking about. Epinephrine and glucagon will activate hormone-sensitive lipase. So the hormone-sensitive lipase is activated when it is phosphorylated. So the kinase, the protein kinase A, will activate the hormone-sensitive lipase. And this is, if we go backwards, protein kinase A is activated by the CAMP. CAMP is activated by the adenyl cyclase. And what activates this adenyl cyclase? Two things. We said epinephrine and glucagon. Insulin, on the other hand, we can see it stimulates the phosphatase, the removal of the phosphate. Once you remove the phosphate, this inactivates the hormone-sensitive lipase. And then this is just simply the metabolism of glycerol. So we said the glycerol will dissociate from those three fatty acids and will go to the liver, where it can be used as an intermediate in gluconeogenesis. And it's two simple reactions. A glycerol kinase, it will phosphorylate the glycerol and basically make it a glycerol phosphate. Then to another step, we'll finally get glyceraldehyde 3-phosphate. And glyceraldehyde, I think you guys have heard this, as an intermediate in gluconeogenesis and glycolysis. So this is another way we can use triglycerides as a source of energy, besides the free fatty acids. So, okay, so this is the main bulk of our talk, inshallah. So, basically, to start off, as we said, we mobilized, now glucagon has done its effect. We've broken down the free fatty acids using those three lipases, the adipose triglyceride lipase, the hormone-sensitive lipase, and the monoglycerol lipase. Now the free fatty acids have mobilized, have went into the circulation, and have entered any cell. It could be any cell, but let's say the liver, for example. In the cytosol, what happens is, or first and foremost, we want to oxidize these fatty acids. The oxidation takes place inside the mitochondria. Now, but how does this happen? How do we get from the fatty acids into the mitochondria? There's numerous steps. It has to be activated first and foremost. So we need to activate this fatty acid. So the very first step, using an ATP, we're adenylating it. So we're using energy to activate this fatty acid. It's very important. And using this ATP, as we'll see in the next step, makes this step irreversible. Okay. After that, we have the addition of coenzyme A. So now we first of all, we activated the fatty acid with an ATP. And then we added a CoA on top of this. So now we have fatty acyl CoA. Now the fatty acid is finally activated and it can enter. However, it needs something to be attached to it in order to go through both membranes of the mitochondria. This is what we call carnitine. A carnitine will attach to the fatty acyl group and will allow its transportation through, as we'll see in the next few slides, through two transporters, CPT1 and CPT2, which are present on both membranes of the mitochondria. So carnitine is very important because without this, the long-chain fatty acids will not be able to enter. Okay, so the carnitine attaches to what we call long-chain fatty acids and will allow their entering through the two mitochondrial membranes. So now we've went through the first steps in the cytosol. We added an ATP, we activated the fatty acid, we activated it by adding a CoA, and now we also added a carnitine in order to allow it to enter the mitochondrial membrane. So after it enters the mitochondrial matrix, what happens is CPT2, we'll talk about this in depth, don't worry about it, will remove the carnitine and reattach the CoA, and the carnitine will go back to the surface and bind to any other fatty acids. So it's a recirculation. So the carnitine will bring in a fatty acid, it will be cleaved off by CPT2 and will turn and bind to another fatty acid. So now we have our fatty acyl CoA has finally entered the mitochondria. So it's ready for beta oxidation. Beta oxidation is 4 series of reactions, where the first reaction is what we call a dehydrogenation reaction, essentially removal of a hydrogen. So this hydrogen will attach to FAD. FAD will turn into FADH2. So we removed a hydrogen and it attached to FAD. The second reaction was a hydration reaction, which is essentially just adding water in the fatty acyl CoA. The third step is another dehydrogenation reaction. However, this time it is NAD, which is where the hydrogen will be attaching to. And then the final step is thiolytic cleavage or cleavage. Essentially, we get 2 products. We get the acetyl CoA, which is the main goal, and a fatty acyl CoA, which is 2 carbons less. So it's 2 carbons shorter, and essentially it will return to the very first step right here. I will go through these series of steps again. So if we had a fatty acyl CoA that is or a fatty acid 16 carbons in length, after these 4 reactions, it is now a 14 carbon length fatty acid. And it will return and go through the same series of reactions again. It will become a 12 carbon fatty acid, and will go again and become 10 carbons, and so on and so forth. And each time you're creating an acetyl CoA as a result, which is very important. And we're going to be talking about how this acetyl CoA is going to be used in the production of ketone bodies. Does anyone have any questions on the slide? We'll be going in depth, inshallah, into each part in the coming slides. But if you have any questions so far, don't hesitate to ask. Okay. What is thyroid cleavage? Thyroid cleavage is essentially the cleavage of, we'll talk about this in more in depth, essentially is cleaving 2 carbons to create an acetyl CoA, and essentially making a fatty acetyl CoA 2 carbons less, because we want to cleave essentially the acetyl CoA away from the fatty acyl CoA, making it 2 carbons less, and this will return. And this is how we essentially reduce the amount of carbons in a fatty acid. No worries. So, okay, another question. So the net acetyl CoA is 8 if the number of carbons is 16. Exactly. So this whole process is, no. This is the thyroid cleavage is the 4th step in this beta oxidation. So the first step is dehydrogenation. Second step is hydration. Third step is again dehydrogenation. The 4th and final step is thyroid cleavage. It is not the entire thing. Okay. And whoever asked, yes, the net acetyl CoA is 8. Yes, if you have a 16 carbon fatty acid, correct. So this is again showing you basically the first step, as we said here, the activation of the fatty acid, which is very important. We see here the fatty acid is activated before going into the mitochondria. It requires ATP to be activated. And this ATP, after being used, will yield 2 PPIs. Right? I'll show you in depth here. So we have the fatty acid right here. We added ATP and a coenzyme A. As a result, we use this ATP, and as a result, it becomes an AMP and 2-phosphate, a pyrophosphate. So we use the energy from the ATP, and it gave us an AMP and a PPI, or a pyrophosphate. This pyrophosphate is immediately hydrolyzed, immediately degraded into 2-phosphate. and this causes it to be irreversible. So we use an ATP. We want the long chain fatty acid to be activated and to enter the mitochondria. If this step wasn't here, it could have been reversed, but we want this step to be irreversible. We want to activate the long chain fatty acid and we want it to enter the mitochondria. If this step is not irreversible, it can, be reversed and will inhibit this process. So once we use this ATP and it's hydrolyzed into AMP and pyrophosphate, the pyrophosphate is quickly hydrolyzed to yield 2 phosphates, and this makes it irreversible. I hope this makes sense. Did everyone get this? Does it make sense? So again, the enzyme used for this reaction is a long-chain fatty acyl CoA synthetase. Okay. So essentially, we're talking about carnitine right now. The two transporters, you could say, in the mitochondrial membranes that are required to enter for the fatty acid to enter into the mitochondria. So we have what we call CPT1 and we have CPT2. So in the cytosol, what happens is the acyl CoA, we've created the acyl CoA, but we need carnitine to be added onto it in order to enter the membranes. We said carnitine is needed to enter the mitochondrial membranes. So what CPT1 does initially, it essentially replaces the CoA with carnitine. The CoA is replaced with carnitine. Both the CoA and the acyl, now the fatty acid with carnitine on top, will both enter through CPT1. They will both enter, we see acyl carnitine and the CoA will both enter. And the carnitine will essentially be cleaved in the CPT2. CPT2 will essentially cleave it after it's entered the mitochondrial membrane. After it's entered the mitochondrial matrix, the CPT2 will cleave this carnitine and will be reused for any new free fatty acid. So we'll repeat this one more time. So we have a fatty acyl CoA. We've basically activated it using ATP, and we made this receptor irreversible by hydrolyzing the pyrophosphate. And we've added a coenzyme A onto this fatty acyl CoA, or fatty acid, essentially. And once it reaches the mitochondrial membrane, CPT1 will add a carnitine instead of the CoA. And with the carnitine, it can now enter through the two membranes into the mitochondrial matrix. So once it has entered the mitochondrial matrix, it has went through the first membrane into the second membrane. What happens here is that CPT2 will cleave the carnitine and will reallow the CoA to reattach onto the fatty acid or fatty acid in general. So essentially the carnitine is being reused. So it brings in a fatty acid cleaved by CPT2 and is reused for any new fatty acid. Isn't carnitine already added before coming to CPT1? The CPT1 itself is what adds the carnitine onto it. It adds the carnitine onto it, okay? The CoA already was there. Yes. So Noura, the CoA was already on the fatty acid. The carnitine replaces it, okay? So it can come into the matrix. Once it comes in the matrix, the CPT2 will remove this carnitine and CoA will reattach. So it's not going anywhere. It's just to allow it to enter the matrix. So the CoA is very essential, of course. CoA is very important. However, to enter the mitochondrial membrane, it needs carnitine. So carnitine will take its place. And once it enters the two matrix or the mitochondrial matrix, the CPT will remove the carnitine and will add CoA back on, essentially. Yes. Whoever asks this, carnitine is only necessary for long-chain fatty acids. It's a very important concept. So this whole process of activating, of CoA addition, of essentially carnitine, the usage of carnitine. This is specifically for long chain fatty acids. So if this was a medium chain, which is around 8 to 10 carbons, it will enter the mitochondria, no problem. Doesn't need carnitine, doesn't need activation. It will enter freely. However, this process is only for long chain fatty acids. Okay. Okay. So why are we talking about CBT1 and CBT2? So the clinical significance of CBT, let me see, no problem. So CPT1, the clinical significance is that there's something called carnitine deficiency. So primary carnitine deficiency is basically where you have symptoms of cardiomyopathy, skeletal muscle weakness, and hypoglycemia because you cannot mobilize or bring in long chain fatty acids into the mitochondria. You cannot use beta oxidation on these fatty acids in general. Essentially, CPT1 is inhibited by malonyl CoA. This is just something essentially because malonyl CoA is, I think you guys did this yesterday with the fatty acid synthesis lecture. Malonyl CoA is in the intermediates of synthesis. Okay, so during synthesis, you do not want fatty acids to be degraded. So you have just created a fatty acid. You don't want it to be created or degraded immediately. So you want the fatty acid to go to the adipose tissue, to be stored, to be used in other places. So if malonyl CoA did not inhibit CPT1, most or some of the fatty acids that are just being synthesized will already be degraded, which is of course not what the body wants. So therefore we have an inhibitory or a negative feedback system, you can say, where the malonyl CoA inhibits degradation of fatty acids by inhibiting CPT1. Did that make sense? Okay. So CPT2, there's something called CPT2 deficiency, and this is specifically the myopathic form. So as we said before, the CPT2 removes the carnitine and prepares the fatty acid for oxidation. And most teachers, of course, have this, but the most common defect is just in the muscles. So what you have is the doctors could ask you a question like this: symptoms of muscle aches, rhabdomyolysis, and they could be prolonged or provoked by fasting. Symptoms could be exacerbated by high-fat diet, and a muscle biopsy shows elevated muscle triglyceride as lipid droplets. And primary treatment is just seize muscle activity and give glucose. So essentially, if you see the symptoms, and as I said, on biopsy, you have high triglyceride, you know it's a CPT2 deficiency. Simply, the doctor could ask you this question, and you should know the deficiency and the symptoms involved in this disease. So going in depth into beta oxidation in specific. So we said the first reaction, which is dehydrogenation, or in other words, oxidation, is removal of the hydrogen. And this basically gets added onto FAD, as we said. And then we have hydration, the addition of water into the fatty acyl CoA. And then we have another dehydrogenation reaction, where essentially we're removing another hydrogen, but this time it's getting added to an NAD. And then we have thiolytic cleavage into two products, as we said, acetyl CoA and a fatty acyl CoA, which is 2 carbons less, and will get repeated through the circulation. I don't think we did. There's a deficiency of. So there's no. You guys haven't taken CPT1 deficiency. This is just carnitine specific deficiency specifically. So the primary carnitine deficiency is a genetic disorder of cellular carnitine transporter system and appears at age of 5. These symptoms are specifically for carnitine, not CPT1. I think I confused you guys over here. So this is basically just talking about CPT1, its significance. It's associated with these two diseases. And malonyl CoA inhibits CPT1. So I think this is the main point of this slide, the malonyl CoA inhibiting CPT1, but there's no disease involving CPT1 specifically. I'm sorry if I confuse the guys over here. But this is the disease you guys should focus on, CP2 deficiency. So again, these are the 4 steps. And after these 4 steps, the fatty acyl-CoA will be 2 carbons less, and will re-enter the reaction again, and will be repeated until we get a 2 carbon or a final acetyl-CoA. That's what we get. We get essentially, if we had a 16 carbon fatty acid, it will go through the steps until we yield 8 acetyl-CoAs, as an example. And these enzymes, as you can see, acyl-CoA dehydrogenase, and all the, I don't think the doctor emphasizes on them. He didn't emphasize them with us. So please make sure if he does emphasize them. However, yes, so it will go the amount of cycles until you yield, basically you break down the fatty acid until no carbons are left, until no fatty acyl-CoAs are left, and you get production of acetyl-CoA. So the acyl-CoA dehydrogenase family is called the family for a reason, because there's different enzymes for different length of fatty acid chains. So you have long chain acyl-CoA dehydrogenase, you have medium chain, as we're going to talk about, and you have short chain. No, it's not important to know the cycles. It's just a concept, So if each cycle you remove 2, it will be, it's just a concept. It's just, if you had, let's say 14, you can have 7 cycles. If you have 12, you can have 6. It's just because this is a concept that every cycle you're removing 2 more carbons and the cycle will get repeated. So now we're talking about medium chain acyl-CoA dehydrogenase deficiency. So I think this is a very important disease in your lecture. Essentially, you cannot hydrolyze or make use of the medium chain fatty acids. So what happens is you have a non-ketotic hypoglycemia. So what does it mean by non-ketotic? Can anyone answer me? What does it mean by non-ketotic hypoglycemia?
Acetyl-CoA is not produced because the production of acetyl-CoA is stopped after you reach the medium chain fatty acids. So because there's not enough acetyl-CoA, the ketone bodies aren't formed.
Great. So who answered this? What was your name?
Zefa.
Perfect answer. So if you guys heard him, he said it's non-ketotic hypoglycemia because we cannot produce acetyl-CoA. So let's say we had a long chain fatty acid and it entered the mitochondria after the activation steps and has went through beta oxidation. Once it reaches 10 carbons, there's no enzyme to further beta oxidate the fatty acid. So we cannot break it down to acetyl-CoA. So we're going to have a lesser amount of acetyl-CoA, and as a result, we're going to have a lesser amount of ketone bodies. And this is evident during fasting. So you'll see them have these symptoms. You're going to have profound fasting hypoglycemia, because when they're fasting, they cannot make use of free fatty acids, and low to absent ketones because they cannot produce acetyl-CoA, as we said. And of course, these other symptoms, lethargy, coma, and death if untreated, is very severe disease. And this is also very specific. See 8 to 10 acyl carnitines in the blood. So what does this mean? It means that the long chain fatty acids are entering the mitochondria. They're going through beta oxidation, but once they get to carbon 8 or carbon 10, they're done with their oxidation. They're not further oxidized. So you will see them in the blood. They may be provoked by overnight fast, and treatment is with IV glucose. And just we can see prevention here. Frequent feeding, high carb and low fat. So it makes sense because you don't want to give them fat if they cannot make use of it. So that you should make sure that they have a high carbohydrate diet and low fat diet. we're going to be talking about now peroxisomes. So I think it's very simply just one concept you have to understand. Peroxisomes, as you guys already know, have catalase. So essentially, the beta oxidation can also occur in the peroxisomes. However, the difference is instead of this enzyme, as we said, the acyl-CoA dehydrogenase, it's the same enzyme, same as this enzyme right here, acyl-CoA dehydrogenase in the first reaction. Essentially, instead of adding the hydrogen onto FAD, it will add it onto H2O and will create, or O2, sorry, onto oxygen, and will create what we call H2O2. And catalase will then basically remove or break down this hydrogen peroxide, H2O2, into water and oxygen. So that's all it is. The difference is that the peroxisomes, they will basically, during the dehydrogenation reaction, it will put the hydrogens onto an oxygen, creating hydrogen peroxide. Of course, hydrogen peroxide is very acidic. We don't want that in our body. So then the peroxisomes have the enzyme catalase, which will convert the hydrogen peroxide into water and oxygen. So that's all you guys have to know from here. The usage of catalase and the addition of the hydrogen atoms onto oxygen instead of FAD. Okay, so all we've been talking about so far, 16 carbons, 14, 18, they will all produce acetyl-CoA, and it's an even amount of carbons. Therefore, you will end up with a certain amount of acetyl-CoAs. If you had 16 carbon fatty acids, you're going to have 8 acetyl-CoAs. However, what if you had an odd chain fatty acid? If you had 17 carbons, you're going to go through the reaction. You're going to go 2 less. You're going to have 15. You're going to have 13, then 11, 9, until you reach 3. Of course, we know that acetyl-CoA is 2 carbons, not 3. So what you have as a result is what we call Propional Coe. So Propional Coe has to have a certain reaction to basically utilize these odd number fatty acids. So through 2 enzymes, it's converted from Propional Coa into methylmalonyl Coa and then succinyl Coa. You guys have heard of succinyl Coa before. Where have you guys heard this before? You guys remember where succinyl Coa is? Yes, indeed. Yes, everyone's correct. So it can be utilized in the citric acid cycle. This is very important because now Propional Coa can be utilized because we had an odd chain. We cannot make another acetyl Coa, so we have to use Propional Coa in another fashion. So the first reaction uses something called Propional Coa carboxylase, and this requires biotin. I remember they emphasized on the requirements of the enzyme. So this enzyme requires biotin. This requires B12. You guys should remember this. And simply the enzymes follow the name of the substrate. So Propional Coa is a Propional Coa carboxylase. Methylmalonyl Coa is a methylmalonyl Coa mutase. And this one requires biotin, and this one requires vitamin B12. So just remember those 2 facts. So now we're going to talk about formation of ketone bodies. So we have done all our steps. We started off with mobilization of the free fatty acids. We use all our lipases. Now the fatty acids were activated and they entered the mitochondria. After entering the mitochondria, they went through beta oxidation. And the 16 carbon fatty acid has now produced 8 acetyl-CoAs. So what do we do from now? These acetyl-CoAs can be used to make ketone bodies. Now, ketone bodies are very important. They're an energy source that can be utilized by enzymes that have or organs that have an enzyme called thiolase. 2 organs have this: muscles and the brain. So this is very important because the brain mainly uses glucose. But if you're in, let's say, a mode of starvation, you've been starving for 20 hours now. That means you haven't had food for 20 hours. That means there's no more glucose from the or carbohydrates from the food. You've used up all your glycogen storages. You've used up the maximum capacity of gluconeogenesis. So now the free fatty acids are mobilized and they need to be used to form ketone bodies. and ketone bodies can be used as an energy source for the brain. So it's very important because in a mode of starvation, you can survive for very long times because there's this mechanism involved. So first and foremost, okay, so after all these enzymes, after all these processes, I think there's a question. Can you repeat the last slide? No problem. Okay, so simply Propional Coe is the result of beta oxidation of a odd chain fatty acid. So odd chain, meaning it is 17 carbons, 15, 19, whatever carbons it is, it's an odd chain. So remember, as we said, the beta oxidation removes 2 carbons at a time. So each reaction, each full 4 enzyme reactions, it will yield acetyl-CoA and an acetyl and a fatty acyl-CoA, which is 2 carbons less. So you have acetyl-CoA and a fatty acyl-CoA, which is 2 carbons less. So if we started with a 15 carbon, we reduce it to 13 carbons, and we keep going until we reach 3 carbons. 3 carbons is Propional Coe, is not acetyl-CoA. So we need to utilize this. The way we utilize this is conversion of Propional Coe into succinyl-CoA. Succinyl-CoA we know is in the citric acid cycle and can be used for utilization of energy in the citric acid cycle. So this is just the usage, how you use odd chain fatty acids, essentially. So the Propional Coe would be converted into methylmalonyl CoA, and then from that into succinyl CoA. And the succinyl CoA will enter the citric acid cycle. That's simply all it is. Did that answer your question? I think it was Rodwan, I think. Okay. So, as we said, formation of ketones is very important because we can utilize this as an energy source in modes of starvation. So we have the fatty acyl-CoA, and after beta oxidation, we're going to form acetyl-CoA. So we're going to combine 2 acetyl-CoAs, and this gives us something called acetoacetyl-CoA. Okay. And by HMG-CoA synthase, this creates HMG-CoA. And then HMG-CoA will then be converted by HMG-CoA lyase into acetoacetate. This is the very first ketone body. And then acetoacetate can be converted into 3 beta hydroxybutyrate and acetone. So those are the 3 ketone bodies that can be formed. So as we said, the first step was formation of acetoacetyl-CoA, and then conversion into HMG-CoA by the HMG-CoA synthase, and then HMG-CoA is converted into acetoacetate, and then from there on, it's converted into the other 2 ketone bodies. Essentially, the differences, I honestly can't really tell you. It's just a simple reaction. Yes, whoever asked, acetoacetyl-CoA is essentially 2 acetyl-CoAs combined. So we need 2 acetyl-CoAs. They're combined to make acetoacetyl-CoA. Now, HMG-CoA is just the addition of HMG onto the acetoacetyl-CoA. You're removing an acetyl-CoA and making adding an HMG. I don't think this is very important. It's not a significant. I don't think they're going to ask you on this. It's not very important. However, just know the steps of reactions. I think that's the most important thing to emphasize on. And of course, that this HMG-CoAs synthase, is the rate limiting step in the ketone body formation. Okay, so now we formed ketone bodies. Now we have to utilize them. The only organs that can utilize them, we said, are muscles and the brain. No, there's 3 ketone bodies: acetoacetate, 3-hydroxybutyrate, and acetone. They're all 3. But this acetoacetate can convert into the other 2. It doesn't mean that this one's not a ketone body. It just can be converted into the other 2. Okay, so we created now a ketone body, either acetoacetate, acetone, 3-hydroxybutyrate, all of them. So now how is it utilized? So that's a very important thing because we need to utilize it for energy. So what happens is the acetoacetate or the 3-hydroxybutyrate are then taken up by the muscles or the brain and are converted back into what we call acetyl-CoA through the opposite reactions. And basically what we have is that these have what we said, thiophorase. Thiophorase, I think I said thiolase, my mistake. I meant thiophorase. The enzyme that is in the muscles and the brain is thiophorase, and it can convert acetoacetate into acetyl-CoA using succinyl-CoA and the enzyme thiophorase. So what happens is right now, we have acetoacetate and we have 3-hydroxybutyrate. Now, these go into the muscle and are converted back into acetyl-CoA. Remember, acetyl-CoA is 2 acetyl-CoAs, and it's converted back through the enzyme thiophorase. This is only present in the muscles and the brain. So it's basically an opposite reaction. We're taking the ketone bodies made in the liver and we're converting them back into acetyl-CoA. And this acetyl-CoA can be converted or used for energy in the citric acid cycle or any other places. So the 3 ketone bodies are simply acetoacetate, 3-hydroxybutyrate, and acetone. Did anyone not understand this slide specifically, how the ketone bodies are being used? Okay, no problem at all. So basically what happened is we are now finished with the beta oxidation. We have broken down the fatty acids and the acetyl-CoAs are now being used to form ketone bodies. And through these 4 reactions, as we said, or a couple of reactions, it will be converted into the 3 ketone bodies, which are acetoacetate, 3-hydroxybutyrate, and acetone. Now, the importance of this is that because 2 places in the muscles and the brain can utilize this as an energy source. And that's because they have an enzyme called thiophorase. So the thiophorase right here, you can see. I want to highlight this specifically. So let me see this. Anyways, the point I'm trying to make is that the thiophorase is an enzyme that can reverse this reaction. So we have formed the ketone bodies in the liver. They get transported through the blood into the muscles or the brain. And thiophorase can reverse this reaction. So we had ketone bodies. We don't want ketone bodies. We want acetyl-CoA. So it reverses this and reverses the reaction and forms acetyl-CoA. And then that will be converted into the 2 acetyl-CoAs, and that can be used as an energy source. Did that answer your question, Nora? Okay, great. So we finished with all the objectives of this lecture. So we're going to go over a couple questions, okay? Okay, can someone read this question out loud? Anyone wants to volunteer? If not, I can read this myself. So basically, essentially what we want to see here. Parents of a 3-month-old infant arrived at the ER, agitated and frightened by the extreme lethargy and near comatose state of their child. The examination showed the infant to be severely hypoglycemic and low measurable ketones. Remember this point, it's very important. Blood analysis also indicates an elevated in butyric and propionic acids, as well as C8 and C8 acyl carnitines. A deficiency in which of the following enzymes is most likely responsible for these observations? Okay. So why is it not CPT1? Can anyone answer me this? Okay, so we'll go step by step. So the reason why it's not CPT1 is because, yes, I don't understand. Okay, no problem. We'll go step by step. Don't worry. So first and foremost, 3-month-old child, he comes with extreme lethargy. He is severely hypoglycemic. So there's severe hypoglycemia. and low ketones. So whenever we see low ketones, it means there's no degradation or no oxidation of fatty acids occurring, whether that be in any of the following steps. It means there's a problem in oxidation of fatty acids. We cannot produce acetyl-CoA. And then it says an elevation in butyric and propionic acids, as well as C8 acyl carnitines. So C8 acyl carnitines are a medium chain fatty acid. So it's saying that they are elevated. It means that they're not being degraded. C8 acyl carnitines are medium chain. Therefore, because they are not being degraded, we can say that there's a deficiency in the medium chain acyl-CoA dehydrogenase. Yes, perfect. Because whoever answered this, both of you two, it can be A because there will be accumulation of long chain. It wouldn't be accumulation of medium chain at all. So that's perfectly correct. And yeah, did that answer your question, Nora? Did you understand why it's E? Okay, great. Okay, next question. Okay, so if C8 wasn't mentioned, could it be CPT1? Well, okay, so I'll tell. Well, the symptoms are not specifically. We haven't done CPT1. CPT1 was not a deficiency. It was not a disease taken. However, if we were thinking about it, there would have to be an elevation of long chain. They did not say long chain or long chain fatty acids are being elevated. They did not say anything similar to this. The reason why we thought it was medium chain is because we saw that there was low ketones, so it's a non-ketotic hypoglycemia, and there are C8 acyl carnitines. So that means there's medium chains that are not being broken down. If it was a CPT1, we'd have an elevated long chain fatty acids in the blood. So essentially, in this answer, it has to be. Okay. So in this question, an individual suffers from a defect in one of the enzymes required for the synthesis of carnitine. If this individual does not have adequate intake of carnitine in his diet and is fasting, which of the following would most likely be the most likely observation in this person compared to conditions of normal carnitine intake? Yes, we use two acetyl-CoAs and fatty acid to form ketone bodies. Okay. If you have a question, don't hesitate to ask. No problem. Okay. So does anyone have the answer for this question? Can anyone answer this? So Mohammed is saying A. Mohammed, can you read question answer A specifically? What does it say? It says, let's read this together. So Mehdi is saying B. Okay. It could be A or B. So why would it be B? Why would it be A? Who wants to argue? Okay, yes. So I think you guys have got this. So A is not even involved in fatty acid synthesis. Dicarboxylic acid is not a thing with fatty acid oxidation. So it will not be involved in this process in the first place. So it would not be elevated. So B, it would be elevated because we cannot bring elevated or long chain fatty acids into the mitochondria. So they will be elevated because we cannot break them down. Did you understand this? Did everyone, is this clear? Hopefully this is clear. I know it's confusing because it says long chain dicarboxylic, but be very careful with the specific words because that is not a fatty acid. Okay? They like to trick you guys with these questions. Okay, I think we can move on to the next question. So during prolonged starvation, the brain increases its use of which of the following substrates in the blood as an energy source? Okay, great. So I think everyone understood this. We use ketone bodies. The brain uses ketone bodies in a mode of starvation. In a mode of starvation, the brain can use the ketone bodies as a result of having the enzyme thiophorase. So specifically because it has this enzyme, we can utilize these ketone bodies as a result. Okay, another question. A 6-year-old boy with progressive muscle weakness is found to have a systemic defect in the production of carnitine. Okay, so therefore this child has a limited ability to produce ATP in skeletal muscle because of a defect in the degradation of long-chain fatty acids via beta oxidation. Which of the following is most likely result of this defect in muscle? Okay, great. I think the concept is very clear. We already talked about this before. If you have decreased carnitine, you're going to have decreased degradation of long-chain fatty acids. That means it will be accumulated in the adipose tissue. There's going to be increased storage or increased stores of triglyceride. This will not affect cholesterol. This will not affect glycogen, of course, or lactate. So yes, everyone is right. The answer is D. Last question. A 2-year-old boy is admitted to the hospital because of generalized weakness, repeated episode of vomiting, and coma after several days of reduced food intake because of a minor febrile illness. He is treated successfully with IV glucose, and after 13 hours of fasting, when the child is well, his serum glucose and beta-hydroxybutyrate levels are low, while free carnitine concentrations are increased. Triglycerides containing only medium-chain fatty acids are fed, and the serum beta-hydroxybutyrate concentration increases to the reference range. Which of the following disorders of fatty acid metabolism is most likely diagnosis? Okay, I like the variety in questions or answers. Okay, so we'll take this step by step because it's a very kind of a confusing question. So first of all, the child comes in after fasting. He reports his symptoms of weakness, vomiting, coma after fasting because of minor febrile illness. He's successfully treated with IV glucose, and after 13 hours of fasting, he has low glucose and low beta-hydroxybutyrate. He has low ketone bodies. Okay, so he has a hypoketotic, a low ketotic hypoglycemia. And also what it says is the free carnitine concentrations are increased. Triglycerides containing only medium chain fatty acids are fed, and the serum beta-hydroxybutyrate concentration increases the reference range. So this is the most important line right here. Triglycerides containing only medium chain were fed, and then the serum beta-hydroxybutyrate was increased to the reference range. So what happened is this patient, this child, and he was fasting, and they checked his glucose and his ketone bodies, and they were low. They gave him medium chain fatty acids, and the ketone bodies increased. So what does this mean? This means that medium chains are, they're okay, they're oxidized. That means that the child can utilize the medium chain fatty acids. When they were given, the ketone bodies increase in the body. So that means that the medium chain acyl-CoA dehydrogenase is working. It's not deficient. It's working because we gave him medium chains and he utilized them. So the difference here is that what happens is the answer is actually A, CPT1 deficiency. Why? because two things. He cannot make ketones unless they're medium chain, because what we said before is that medium chains can enter the mitochondria simply. They can simply diffuse. However, long chains, they require carnitine. So if you don't have carnitine, you can't degrade long chains. So if your diet consists of medium chain, you can still use it. You don't need carnitine. However, long chain, if you have a CPT1 deficiency, you're not able to utilize them. So you're going to have low ketone bodies. Okay. And also here we see there is free carnitine concentrations are increased. Yes, I'm sorry. This question isn't really with you guys. It's just a concept. I'm sorry, I don't think this would ever come because you would never ask about CPT1 deficiency. However, as a concept, just to understand that carnitine, yes, he cannot utilize long-chain fatty acids. Why not B, dietary carnitine deficiency? Because essentially what happens here is that we can see his free carnitine are increased. Doesn't have a deficiency in it. So B would have the same symptoms. You're completely correct. Whoever said B, I think it was Rodwan. B is completely correct in terms of the symptoms. However, in this line it says free carnitine is actually increased. So it's not deficient. So it's increased actually. So the answer is actually A. I know it's a very confusing question. This is very difficult. So do not worry about it if you didn't get it. But just as a concept, just to understand that long chains need carnitine to enter, while medium chains can simply enter the mitochondria. Yes, Abdurrahman. So the long chains need carnitine to enter the mitochondria. Any more questions? No worries. So everyone, if you guys have any more questions, please don't hesitate to ask right now. And I'll put my email in the chat just in case anyone wants to ask privately. So any more questions? I'm so sorry. I read it wrong. Is it Rodan? I don't know how to pronounce that. Sorry. If there's no more questions, I think we can end this